So I wanted to start with talking about what happens upon an infection, because that seems to be one of the biggest problems we have with ME/CFS. And one thing that happens is that the mitochondria are fragmented. That's in a recent publication. There's also oxidative stress is activated, and ATP production is reduced. So when you do get an infection, you feel tired. That's most likely because the ATP production has been reduced. So first I want to talk a little bit about the oxidation. It's something that's known, but it hasn't been really investigated very much in ME/CFS. So this work is done by Vishnu Shankar and Mark Davis. My contribution in this work was that we supplied the ME/CFS samples. This happened during the pandemic. The study was done during the pandemic, and these were all from frozen samples. We usually like to use fresh samples, but patients did not want to come into the lab. However, Stanford had a rule that if you worked on long COVID, you could work in the lab, and so Vishnu did some work with long COVID as well, as simultaneously with doing work on ME/CFS. So you see a large number of reactive species. Not being a chemist, you probably don't pay much attention to this, but all these molecules are very reactive and can react with other components in your body. So looking at the level that's present, and this is both ME/CFS patients and, as I just mentioned, long COVID patients. And what you see from this compared to healthy controls is that there is a lot of oxidative reaction with ME/CFS, and to some extent a lesser so with long COVID. And this is looking at different cell populations, and you see that in all cell populations. Another thing is a little bit of surprise. If you look at male versus female, the females have a lot of oxidative reaction, and the males less so. I have no idea why that's the case. But I want to talk about, in fact, I'm going to talk about a couple of small molecules that I think need to be studied in ME/CFS and haven't been studied very much. One of them is a compound called BH4, and because of the oxidative stress, it's likely to be low. I'm not going to show you the structure of it because that's not going to help, but this compound is the core structure of this compound looks very much like folate, and it's a complicated molecule, very, very similar. And the H4 is because there are 4 critical hydrogens in that molecule, and it's a reducing agent, which means it can reduce the oxidative stress to some extent. And when it gets reduced, or when it gets oxidized, it goes to 2 hydrogens, and they call that BH2. There are some compounds that can be made which are called BH3. The problem is that studying this molecule is hard, and that's probably why it hasn't been done very much. The hard part of it is it's hard to quantitate. How much do you have? And it's usually done by a mass spectrometry instrumentation, but it's hard to get good quantitation with a mass spectrometry instrument. It's sensitive. There's also not very much of this, so sensitivity is a real problem. So we have been working for about a year trying to find a good quantitative method for seeing that molecule, and it's not very expensive. And so I've had 2 of my really great research associates working with me for years take on this problem. One's a chemist and the other one's a biologist, and they worked together. So I want to show you a little bit about why this molecule is important. And these are some of the reactions. Most people probably have heard of phenylalanine. It's an amino acid, a critical amino acid. It has some problems if you can't metabolize phenylalanine, certainly after your birth, and you're not metabolizing it very well. It will cause brain retardation, and it's called PKU. And this molecule is converted by an enzyme and BH4 to tyrosine, which is another amino acid. And then tyrosine is converted again with BH4 and an oxidation to DOPA. Now that might start to feel familiar because DOPA is used to make dopamine. And that's a very critical hormone. But this BH4 is also used to convert tryptophan to a precursor for serotonin. This also requires BH4. So you can begin to see that having BH4 levels is probably really critical for your mental state. But that's not all what it does. And it's possibly even more important is that BH4 is used to convert arginine by oxidation to citrulline, but it generates a very small molecule called nitric oxide. Very simple molecule, but nitric oxide is absolutely critical for an awful lot of things in your body. It's used to dilate your blood vessels, for example, but it's used for many things. I'll show you a few of those. One reason why this is such a fascinating molecule is because it's a very simple molecule, and it's very small. And because it's very small, it can diffuse very rapidly. So it's something that's probably developed in early evolution billions of years ago. But also, it can go right through membranes. So that's always a problem for chemistry, in the sense it's hard to get some molecule through a membrane, which is a lipid layer. This just goes right through. So it's a very useful molecule for doing regulation. So those are critical things. And one reason we started doing this study and looking at this is because of a patient. A patient came to Hector Bonilla, who runs the clinic at Stanford for MECFS, and said that she wanted a new prescription for Abilify. And he sort of said, why? And she said, it really, really helps me. And so not just ignoring her, he actually gave her a prescription for Abilify, and then started to use it on other patients and see what would happen. He wound up giving it to 101 patients, and when they came back, he assessed how they were doing. And my research associate, Laura Crosby, was interested in this too, and she compiled all of the data and looked at it and found that about 3/4 of the patients seemed to be really good responders. Not everybody seemed to respond. But it is actually a very good drug for MECFS, I think. A lot of people feel very much better about it, and my son has taken it, and it really, really helped him. Well, that led us to saying, well, what does that do? Well, it's believed that it increases dopamine. Going back to the DOPA, there may be a problem there. And that problem could be BH4. So that's what led us to look at BH4. And let me look a little bit at the nitric oxide. And these things I just mentioned to you. But one of the critical things as well is that nitric oxide can react, and it can react with proteins, and in doing that, it modifies their function. So one place where it does that modification, which also may now be critical, is that you have some metals that are very important for parts of enzymes. And those metals are iron, manganese, and copper. And of course, people often know that iron is an important thing, and that's, it's in the red blood cells, but it's used for lots of other reactions. And it will not go through cell membranes. It's charged, it will not go through. You have to pump it into cells. And it's pumped into cells by a metal transporter. But that metal transporter doesn't do anything unless it's reacted with nitric oxide first. And that's where that nitric oxide reaction is critical. And that reaction, of course, to make that requires BH4. I'm going to show you a little bit. These metals are used to scavenge some of these oxidative compounds that are made. And now, if they don't get into cells, they don't work. So the reactive species that are generated by infections is a real problem, and it can cause an awful lot of damage if you don't get rid of them. Now I want to show you, this is a bit of a hypothesis, but I think it might help you understand some things. And that is, why does this disease last so long? Why don't you just get over it? Like a cold. You get sick for a few days and you get over it. Well, this might be one of the a possibility. This is just a hypothesis. That you have an infection and you get oxidative stress. The oxidative stress then winds up lowering the BH4. When you have low levels of BH4, it reduces the amount of nitric oxide. If you reduce the amount of nitric oxide, you limit the amount of iron, manganese, and copper that are getting into cells. These compounds will then reduce the oxidative stress, but you now have them low. And so you continue to keep this cycle going, and you can't get out of it. And so this may be, something like this may be going on in the patients where you get a sustained cycle of reactions that are a problem. And the way what you have to do is break the cycle. You can break that cycle maybe by doing something else that will decrease oxidative stress. If you could figure out a way to protect the BH4, but we don't know how to do that. Another possibility is find a drug that will actually produce nitric oxide. So you don't require BH4 to get the nitric oxide. And we're working with a couple of companies that might have some compounds that will do that. To get these into cells, you can take a lot for your blood, so your blood could have a normal level, but it's not getting into the cell and it's not going to help. So we have to find a way to see if we can break the cycle here. But that could be very simple. So we spent about a year trying to figure out how to measure BH4 with my two research associates. And what we did was try to use a simple instrument called a high-performance liquid chromatography instrument. It's not terribly expensive, and it doesn't take up a incredible amount of space, and it's not technically difficult to use. So that's something very useful. But you can have different columns that you can put with it, and different conditions for running it. And so that's what we spent time doing, trying lots of different columns that had high sensitivity detector and lots of different conditions. And that's what's taken it. We did 400 runs on the instrument from samples from patients to look for this. And this is the result that looks clean. What you have to do is get this peak here separated from everything else. And that was hard. And then you had to get this peak separated from everything else when you did your run. This peak means it's BH4 plus BH2. This peak is BH2. So to get BH4, you simply subtract this amount from this amount. And it looks like it's working fine. I won't go into the details here. It's just that we have to compare it to the literature value of how much is in a healthy person, and we get the same amount. So it looks like it's giving us good quantitation. So now what we'll be doing is recruiting a lot more patients to come to the lab, and we'll look at what is the level of BH4 in the patients. And that's probably the first time that's ever been done. Now, BH4 is often not, you don't know how to measure it easily, and another problem is, what do you do with it? The problem is when you do a blood draw, you lose 1% of the BH4 every minute after the blood draw. So that means you have to either stabilize it or you have to have your instrument very close. We're going to do both. So the instrument is actually about 40 feet from where the lab draw occurs. And so we'll do them very quickly. The other problem is you can help to stabilize the BH4 with other reducing agents. But it takes time to do the addition. So we made a contract with Beck & Dickinson to put these reducing agents into the blood tube that they supply, which is under a vacuum and sealed. So now when you do the blood draw, you immediately have the presence of the reducing agents, stabilizing agents, and then we can still go and actually perform the analysis pretty quickly. But it probably will be safe to freeze the samples. So we've done one patient, and that is what we found is the level of BH4 is 39% of the total, and that total is the combination of BH4, BH2, and BH3. A healthy person is 67% of that. So this is about half. And the other thing of interest is the fact that the amount of total here of BH2 is actually higher than healthy people. And that might be a problem. Then there's a ratio of BH2 to BH4 looks like it's a problem. So these studies have allowed us to get qualified to get a compound called Kuvan, which is a synthetic BH2. It's not exactly the same because you can't get a patent on something natural, but it's close enough to function as a biopterin. And we've given a small dose of that to my son, and it made him feel awful. So we just made the ratio of BH2 to BH4 even worse, and that probably is a problem. So we're not sure how to actually get the BH4 back for people. We do know some people do fine on Kuvan. It really helps them. We want to get those patients and look at what their ratios and so far. But we may have to do something to prevent the oxidation and not just try to give a drug. I just wanted to cover something else that I think is relevant and hasn't been looked at before. We know that EBV virus, which can cause mononucleosis, and HHV6 can cause ME/CFS. There's been a lot of studies in that regard. But the question is, what happens with other infections and stressors? What we have found is that other infections and stressors can activate the endogenous EBV and HHV6. So in other words, when you get these viruses, EBV or HHV6, they're with you for life. And they're in resident in some cells and they're suppressed. But when you get a stressor, they can be reactivated. So I'll just show you the here's some COVID samples. Dr. Bergquist sent them from Sweden. And what we find is that these are patients that have the active SARS virus infection. The ones in yellow are the individuals that have EBV. After they clear the virus, SARS virus, it seems that the EBV drops down. This is EBV, this is HHV6, It's a little interesting that in places where you don't have EBV, you have HHV6. It's possible that these people don't have EBV, and that's why you get HHV6 activated. The other virus we looked at is cytomegalovirus, which is also, and it does happen occasionally, but it's much less. EBV seems to be the major candidate. If you put these all together, it shows reading all three of those herpes viruses. And the way we do this analysis is a multiplex way in the sense that we can do 20 different herpes viruses simultaneously. And there's a couple of other herpes viruses where we see an individual having it, but I didn't put those up. But we add these together, we get 95% of the patients have an active herpes virus. So the question is, we know those viruses can cause ME/CFS. So is this what's causing long COVID? And long COVID is possibly the wrong name. This is long herpes. And that's just a possibility to think about, because there's been a huge focus on the SARS virus must be there, or something from the SARS virus is causing this. It might have nothing to do with it other than the fact that it's an infection. Well, is this the only thing that can cause an infection? Or cause an activation, excuse me. These are just the transcriptional reads, just showing you that the virus looks like it's fully activated and making RNA and going through replication cycle and making new virus particles. So, Wen Zhangshao found trauma samples that we worked on with Ron Tompkins 15, 20 years ago. He found them in the freezer. And we have a lot of data on those samples. So these are all autooxidants, mostly, pretty severe trauma. And what we find is that EBV, again, is activated. And we also know that people can get ME/CFS from an autoaccident or other trauma events. This may be why. So this is possibly happens with a lot of different situations which are major stressors. And that could be the cause. When people say they don't know the cause of it, it doesn't necessarily mean you had a viral infection. It could have been something else. That's another stressor that caused it. So I want to do one last thing, and that is, again, when you have an infection, you activate innate immunity, you feel fatigued and tired. The reason for that is something called, I think we've named this the itaconate shunt, and that reduces ATP production. So when you get any viral infection, you get, or anything that activates innate immunity, you get, the itaconate shunt turns on. And I'll just show you that pathway. This is a citric acid cycle where you get most of your ATP. It starts from glucose, which makes pyruvate, and then goes through these compounds. And when it gets over in here, you start making ATP from these reactions. And that's where most of your ATP comes from, is in the citric acid cycle. But what happens when you have an infection is there's a new enzyme is made in your cells that migrates to the mitochondria, and that makes itaconate. So cis-itaconate, instead of going to isocitrate, goes to itaconate. This is not 100%, but it's a large fraction goes to itaconate, and then to other compounds, and eventually back to pyruvate where you started. That bypasses any ATP production, and that's why you feel fatigued. So the possibility, well, we know that the possibility is that this pathway is on in ME/CFS. And it's locked on for some reason, and it may be locked on because innate immunity is locked on. And we're trying to figure out a way to either unblock it or shut down this pathway. So there's a group at University of Utah working for a FDA-approved drug or a natural product that will block this step. And that will, if that's true, then that will reduce remove the lack of energy that people have. And then they're also making they're doing a wonderful job on this. They're also making a zebrafish model. So zebrafish are very easy to work with. They're very tiny little fish, and you can genetically engineer them. So they're engineering them to do some of the chemistry here, and they're doing it so that they activate the itaconate shunt genetically. And when you do that, they swim slow. So we've shown that they've shown that they can fatigue the fish by this pathway. But then that gives them a chance to find how do you unblock it. So that's encouraging. So I think the generally field has made lots of progress, and I'm really delighted to see people trying to work on treatments and think about treatments. That's the right time now to do that. And I'm guessing we'll have a number of reasonable treatments out there, and maybe even some really great ones. So thank you very much.